Good morning everyone. So today's journal club topic is succinate nanomaterial boost tumor immunotherapy via activating cell pyroptosis and enhancing MHC1 expression. So this article was published in January 2025 with in the Journal of American Chemical Society with an impact factor of 14. So following are the terminologies that we should know before understanding this article. So succinate, it is a metabolite involved in cellular respiration Krebs cycle. In this study, it acts as an immune regulator that helps upregulate the MHC1 expression in tumor cells, making them more visible to the immune system. Pyroptosis, it is a highly inflammatory form of programmed cell death triggered by ion imbalance and capsase-1 activation. So this will lead to the cell membrane rupture and release immune stimulating signals, also known as DAMs. Then Gasdermin D, it is a protein that is cleaved during pyroptosis. Its N-terminal fragment forms pores in the cell membrane, causing rupture and inflammation. Capsase-1, it is a protease enzyme that initiates pyroptosis by activating GSD, MD and inflammatory cytokines like IL-1 beta. MHC1, it is major histocompatibility complex, a cell surface molecule essential for presenting antigens to CD8 plus T cells, tumor cells, often downregulate the MHC1 expression to evade immune detection. So succinate nanoparticles in this study will help to restore this expression. The tumor immune escape, this is the process by which the cancer cells avoid the detection and destruction of the immune system by often through MHC1 downregulation or creating an immunosuppressive microenvironment. The immune checkpoint blockade, it is a type of cancer therapy that removes the brakes from T cells. Its effectiveness is limited by low MHC1 expression, which is the studies aim to overcome. The succinate nanoparticles, it is engineered sodium succinate nanoparticles designed and to deliver succinate into tumor cells, inducing pyroptosis and restoring MHC1 expression. The DSPE-PEG modification, it is phospholipid tech coating used to make the nanoparticles water dispersible and biocompatible for the in vivo delivery. Damage associated molecular patterns, it is molecules released during pyroptosis like ATP, HNGB1, calreticulin that alert and activate the immune system. Reactive oxygen species, it is chemically reactive molecules generated during stress conditions. In this study, succinate nanoparticles induce ROS generation contributing to pyroptosis. The tumor microenvironment, it is the cellular environment around the tumors which can suppress the immune response. The nanoparticles reprogram the tumor microenvironment by increasing the MHC1 expression and triggering the inflammation. The effector memory T cells, CD8 plus EM, it is a subtype of the T cell involved in long-term immunity. Their activation in this study indicates a durable anti-tumor immune response. The TRAF3 gene, it is a gene also known to regulate the MHC1 expression. Previous studies shown that its deletion increases MHC1 expression, but this study avoids genetic manipulation by using succinate instead. So the novelty for this study is this is the first study used to use succinate loaded nanomaterials simultaneously trigger pyroptosis and enhance MHC1 expression in tumor cells. It combines metabolic modulation and technology to amplify the anti-tumor immunity. The dual immune activation, pyroptosis and MHC1 upregulation offers a novel strategy for improving the immune checkpoint blockade therapy. Delivering the succinate directly to the tumor cells via nanoparticles will induce apoptosis, pyroptosis, releasing immune stimulating signals, increasing MHC1 expression, making the tumor cells more visible to the CD8 plus T cells, and enhance the anti-tumor immunity and synergize with the immune checkpoint therapy. So the aim was of this study was to design and test the succinate-based nanomaterials that enhance the anti-tumor immunity by inducing pyroptotic cells, cell death, and upregulating MHC1 expression on the tumor cells. So this is the graphical abstract overview. They made the nanoparticles and they made it biocompatible, and then they inserted it into the cells, which will promote the MHC1 expression and also induce pyroptosis to kill the tumor cells. So the materials and methodology, the succinate metabolite is zirconium-based metal organic framework as the nanoparticle carrier. Tumor cell lines B16F10 MC38, that is B16F10 is melanoma, MC38 is colon carcinoma, and in mice it is C57BH6 for in vivo tumor experiments. So the antibody and reagents for flow cytometry, emission plotting, ELISA was used. So synthesis of MOF, that is zirconium-based metal organic framework, succinate particles using zirconium and succinate. Characterization of particle was done, surface charge and morphology and drug loading was assessed. The cellular assay was done to assess the uptake, pyroptotic markers, MHC1 expression. The animal experiments was done. The tumor bearing mice was treated with the MOF succinate alone with anti-PD1 antibodies. The tumor growth monitoring and the immune cell infiltration and survival analysis was done. The flow cytometry and immunofluorescence Western blotting and ELISA was done for biological evaluations. So this study, the data was presented as mean and standard deviation. The statistical significance was assessed using the student's test for two group comparison, one way ANOVA for multiple group comparison, Kaplan-Meier survival curves for log rank test, and the significance was less than 0.05. The results, so the MOF succinate nanoparticles were successfully taken up by the tumor cells. It released succinate inside the cells, so this activated the pyroptosis, GS-DMD cleavage and capsase 1 activation, and also it increased the MHC1 expression on the tumor cells. So the immune activation, more CD8 plus T cells and dendritic cells in the tumors was seen. Also higher level of pro-inflammatory cytokines was seen. In the in vivo study, reduced tumor growth and improved survival rate in mice was seen. Also synergistic effects when combined with anti-PD1 therapy was also noted. So the discussion, the succinate acts as both metabolic signal and an immune modulator. The nano carrier ensures tumor specific delivery, avoiding systemic side effects. The pyroptosis and MHC1 upregulation provide a 1-2 punch to tumors. So the pyroptosis will trigger the immune alert, and the MHC1 will help the immune cells to recognize and attack the tumors. So this strategy may help overcome the resistance to immune for tumor to be immune checkpoints So the strength of this study, it is a innovative dual mechanism, both pyroprocess and immune recognition enhancement. So effective in multiple tumor models as studied in colon and melanoma. Also, strong preclinical evidence of synergy with existing immunotherapy was seen, and it also demonstrates a rational design for a nanocarrier material to boost the immune responses. The limitations of the study, it was a preclinical study limited to cell lines and mice. The long-term safety and metabolism of nanoparticles was not fully explored. The effect may vary with tumor types that may have a different immune response in the environment. Also, clinical translation will require further optimization and validation. The study design and guidelines, because it was a both in vitro cell line study and an animal study, it followed the ARRIVE guidelines and the MINCHAR guidelines. So the following guidelines were partially followed. The sample size, total number of animals and how the size was decided was not reported, but rationally was unclear. Randomization was implied but not in detail. Housing and husbandry of the animals were not briefly mentioned. Also, batch reproducibility of multiple batches or replicates were partially mentioned. The guidelines that were not followed was the inclusion and exclusion criteria of including and excluding animals was not mentioned. Also, blinding was not done for of outcome assessors. Also, the protocols and registration, the prospective study protocol was not registered. So that's for the guidance. Thank you.